we will get started. So welcome to October's JETZON call. Once again, we have two excellent talks that have been contributed to us. So the first talk is by Henk Jan Hoving of GMR, and I think I will let Henk Jan introduce his subject himself, but thank you very much for volunteering to talk to JETZON, Henk Jan.
Thank you for the introduction, Adrian. I'll now share my screen with you. I hope this is all visible as planned. So my name is Henk Jan Hoving. I'm a working group leader at the Deep Sea Biology Working Group at GMR in Kiel in Germany. And I'll be talking about biodiversity and ecology of deep pelagic invertebrates. And I'm very happy to present some of the work we do in the working group, which I think is very much in line of the context of the JETZON themes. And we'll be talking about detecting the visible and the invisible. To start with, okay, what do I mean with deep pelagic invertebrates? We basically focus on two groups of fauna. So we're interested in the gelatinous organisms, which are shown here, which include hydromedusae, but also ctenophores and siphonophores. And this group of gelatinous zooplankton is increasingly recognized as pivotal players of oceanic food webs. And various studies in the last years have shown that they're both very important as both predator and prey, and also in circulating carbon through oceanic food webs. But they're also notoriously hard to study because of their gelatinous and delicate nature. So many of these organisms that are shown here, so for example, this large siphonophore, you would never capture in this state in a net because it basically falls apart. So you need in situ observations to show and to document them and to recognize them and to quantify them. The other group of pelagic organisms are the cephalopods, and I've been doing my PhD on oceanic cephalopods and life history strategies, and since then I've been fascinated about them. They're a diverse group of oceanic mollusks, and they have a range of features that makes them very interesting to study. So they have very good eyesight, they are very mobile, they are very good swimmers, they have muscular bodies, some of them. Others only use ammonium to increase their buoyancy and are more planktonic almost. And they're incredibly abundant in the world ocean. So just to give you an example, Clark has estimated that annually as much cephalopods are consumed by sperm whales alone as all human fisheries combined. So this is a huge biomass, but it's very not well studied. And they're also very fascinating in terms of life history strategy, because cephalopods, when they hatch from the egg, they consume a large variety of oceanic prey items, and they grow very fast, so with very high conversion rates. And when they reach maturity, they mate. So they have separate sexes, they mate, and then the female spawns the eggs, and this reproductive cycle only happens once, and this is called semal parity, and then the individuals die. And this makes it very interesting also in the context of transfer and transport of flux of carbon, because this biomass of cephalopods that basically suggests that on an annual cycle there are pulses of carbon, of cephalopod carbon, reaching the ocean floors. But we know very little about it. And this is also a theme of my working group, but we will not go into this today, because today we'll talk more about mesopelagic fauna and processes. So when we talk about detecting the visible, I want to show you the different tools that we use at GMR in the Deep Sea Biology group. So we have a toad. We use toad systems like nets to collect our samples, but as I mentioned, where a lot of gelatinous fauna are too delicate to properly sample, so we also use in situ observations, so different kinds of camera technology. And I'll show you a little bit about a towed camera system that we developed at GMR. We use submersibles, both manned and unmanned. And we use different kinds of seafloor instruments, including coring and landers to perform, for example, footfall experiments. So this is a whole combination of both analytics, observational tools, and also molecular genetic tools that I'll tell you a bit more later on in this talk. We collect lots of video and images, and we combine that in a database that is now known as the Ocean Biodiversity Observation Database. So we try to annotate all video that we collect. And this database now consists of video and frame grabs with more than 130,000 annotations of pelagic fauna that is there for reference. And when we perform our research in the different ocean areas, you use in situ observations to establish biodiversity baselines. And just to give you an example, this is Kioimea. It's a very large ctenophore. This animal is about 40 centimeters large. And during our surveys off Cabo Verde, we documented this animal in 150 meters of water depth, and it was previously only known from the Pacific. So it was described by Matsumoto and Robeson from Monterey Bay, and it's only known from the Pacific. So relatively large animals can be discovered using in situ observations in areas that are poorly studied. But it's not only biodiversity that can be detected using in situ observations, it's also interactions between organisms. So this is a large hydromedusa in the water column that's actually feeding on pyrosomes. And there are all these little fish around this hydromedusa that's either using this animal for shelter, or maybe even feeding on the leftovers that the hydromedusa is creating. But it shows that the animals in the middle of the water column have interactions, and these kinds of interactions cannot be discovered using net sampling. And another example to show that in situ observation can show ecological interactions. Another example that is this midwater fish. It's a midwater scorpion fish that we observed off Cabo Verde. And when I show it like this to you, there's nothing unexpected, but we actually observed it in this position. And these midwater scorpion fish, they hang vertically in the water column. And it shows us that the previous image came very natural to us, because we like to see animals the way we know them, from swimming horizontally in the middle of the water column. But by more and more using in situ observations in the three-dimensional pelagic zone, animals behave differently. And we really need these observations to better understand them, because this animal probably uses its large fins to hang in the water column and to feed there. And it's living on the seafloor, but it probably feeds up into the water column, hundreds of meters of the seafloor. So just to give you some overview of the research themes that we study. So I gave example of biodiversity and distribution data. We combine that with auto ecology. So we also study age and growth and reproductive biology. And we also look at the interactions of animals in terms of food webs, but also the biological carbon pump. And all this is put in the context of ocean change. To give you examples of studies that we performed focusing on biodiversity and ecology of deep pelagic invertebrates, I want to show you the results of a cruise that we already did 6 years ago, I can't believe it, off the coast of Cabo Verde, where we were studying mesopelagic communities and how they were distributed in relation to the oxygen minimum zone of Cabo Verde. So this is West Africa, and there is this the Cabo Verde archipelago is situated here, where we had these different stations where we performed a range of sampling, from in situ observations to net sampling to CTD casts. And these are two instruments that we deployed. One is a MOCNES net sampling system, which is basically can discretely sample at 9 different depths to obtain discrete samples from specific depth. And this is an instrument that we developed at GeoMar. It's a towed camera system for horizontal pelagic video transects in the upper, and we performed these in the upper 1,000 meters of the water column by towing the instrument slowly at 1 knot through the water column for between 10-20 minutes. And it's using a high definition video camera. We could document and record organisms larger than 1 centimeter. And it's very suitable to document gelatinous fauna. And by annotating all the video, we were able to reconstruct vertical distribution patterns of animals in their chemical and physical environment. To give you an example, how we go from observations.
Vous m'avez dit, excusez-moi.
How we go.
Non.
How we go from observations to data. So we annotated all the video, and we clearly could identify certain groups of organisms at least, so different kinds of ctenophores, polychaetes, siphonophores, and then we could make these distribution plots from the surface to 1,000 meters of depth, and we did them during day and night. And when we, for example, look at Solmissus, Hydromedusae here on the left lower corner in E, we could clearly see that this animal is distributed throughout the water column. The oxygen minimum zone is depicted here in blue. And Solmissus also occurs there. And when we look at the fishes, we clearly see that during the day, animals are in the middle of the OMZ and deeper, while at night the organisms migrate up to the surface. Now, this is not a new phenomenon. This is known as daily vertical migration, and it's well documented in the literature. But it's encouraging that we see it here in the video, because it shows us that the patterns that the biological patterns can be detected in our video. When we now look at other gelatinous fauna, for example, Liliopsis, which is a small fluorescent siphonophore, we can actually see that this organism is mainly occurring at the same depth as the oxygen minimum zones. Well, the ctenophore Burrowi has a reduced presence and abundance inside the oxygen minimum zones, and maybe even avoiding it. Well, these kind of data we did for more than 30 taxa. And what it tells us is that we could use these taxa to actually predict, okay, which animals are now benefiting or are actually detrimentally impacted by expanding oxygen minimum zones. Because in a future ocean where there's a larger oxygen minimum zone, we can predict that Burrowi, because it's avoiding this oxygen reduced layer, it may have a reduced habitat in the upper layers and actually push down, while the siphonophore Liliopsis may actually have a large increased habitat with expanding oxygen minimum zones. And this is an illustration how we can go from video to data to predictions on how animals may be impacted by ocean change. The next cruise in 2018, we went to the more coastal deep seas of Cabo Verde, also using the manned submersible Iago on the research vessel Poseidon. And what was very fascinating is that in these coastal regions, there were these very high densities of pyrosomes, these pelagic tunicates. And this is a poor image made with a cell phone, but this is the surface at night of some of the islands, where basically the whole sea surface was full of these pyrosomes that came up from the middle of the water column, from the mesopelagic zone up to the surface. And these are actually, this is an image from Pelagios of a upward migrating pyrosome of about 8 centimeters in size. And by looking into the water column, we actually saw that these animals occur there in huge densities, so many, many pyrosomes. And so we decided to really focus on these animals and to study them, quantify them off Cabo Verde, using a different set of tools. And they're also very interesting because in the Pacific, they're now known to actually expand their range into more northern areas. And they're also referred to as glowing sea pickles because they they make their own light, or they're bioluminescent. But off Cabo Verde, we found actually that they're also very important in the middle of the water column for other organisms. They create substrate of various crustaceans to live upon. So this is a large crustacean that settles upon the pyrosomes, and here are smaller amphipods that were living inside the pyrosomes. We repeatedly saw small midwater fish inside the pyrosomes. And I'm actually wondering how they would get out again. By swimming backwards, perhaps, but this was very interesting. And of course, the well-known amphipod Phronema, who basically uses the barrels of the pyrosomes to lay its eggs and to brood its eggs. So these are different associations that pyrosomes have in the middle of the water column. But what I find fascinating is that these animals create substrate in the middle of the water column. We quantified their daily vertical migration. So their primary depth during the day is between 300 and 500 meters. And during the night, they come in massive numbers up to the surface. And we actually quantified the carbon that is associated with this vertical migration. We also were able, using species-specific primers, we were able to find eDNA of pyrosomes down to 3,000 meters. So their impact on the vertical flux is likely goes fast beyond 1,000 meters. And we also, using seafloor observations with a manned submersible and towed camera, we saw that the carcasses of these pyrosomes were consumed by various kinds of seafloor organisms. And we also calculated the flux associated with these pyrosome carcasses. Now, this was all using animals that are visible. But very often when you do deep sea work, you're looking for a specific organism, but in this huge environment where animals have evolved perfect ways to escape predators, and therefore also to escape from sampling instruments, including ROVs or nets or towed cameras, you very often don't see what you're after. And that's why we also need to look at alternative ways to detect animals. And this I want to highlight using and therefore we now more and more use eDNA in our studies. And I want to highlight one published study of this year where we looked at basically combined biologging and eDNA sampling in the foraging zones of deep diving whales. And we did this off the Azores, which is an oceanic archipelago, which is not wrongly illustrated here. So it's much more in the open ocean, of course, in the Atlantic. My apologies for that. So something must have gone wrong, but it's about here the Azores are located. And through a unique collaboration with the group of Fleur Visser, who tags resort dolphins and Cuvier beaked whales off Terceira, where the resort dolphins forage inshore and the Cuvier beaked whales forage deep offshore. But these animals of these species live and hunt relatively close to each other. So we were interested, so how is it possible that these species live so well together while they prey for the same prey group, which are the cephalopods? So our hypothesis was that there must be differences in diversity in the hunting zones of these two whales. So the group of Fleur, they tagged the whales so they could exactly determine the foraging depth of these whales. And my group, we actually sampled water from the foraging zones of these whales to analyze it for eDNA of cephalopods. And we do so by the principle of eDNAs, that every animal, when swimming in through the water, it releases traces of its body. So either through mucus or through feces or through other body parts, but they're microscopic parts that you can collect by pushing water through filters. And you can extract the DNA from those filtration. You can amplify the DNA, and then using different, in this case, cephalopods specific primers, you can actually detect the community of cephalopods that is in this particular region based on eDNA. You can compare the obtained sequences then with reference databases. And since we've been working on cephalopods for quite a long time, we have lots of specimens, and we have comparative specimens where we also have a barcode to compare the obtained eDNA sequences from. This is a complicated graph, but what I want to show here is tell you that we obtained sequences from 39 different cephalopod taxa. Our hypothesis that there was a difference in diversity between the foraging zones of the whales was not, we had to reject, because there was actually a high overlap in prey community composition between the foraging zones. What was encouraging is that 21 taxa we detected with eDNA are known from the diet from the Azores, but or from other areas. And since there was such an overlap in diversity of the animals, both inshore and offshore, we hypothesized that the squids actually go perform an ontogenetic migration from inshore to offshore. As they grow older, as they reproduce, they move into deeper water. So the resource forages on the juveniles of the inshore living squids, while the cuviers dive deep for the larger reproducing squids. And that's basically the hypothesis that we came up with, that we now need to test with further sampling, physical sampling using nets and underwater vehicles. So with that, I want to finish my talk in this JETZON session. I want to thank the conveners. Thank you, Adrian, for inviting me, and I'm very happy to answer any questions on the mesopelagic work we do.
Thank you, Henk. A fantastic range of stuff. It's really, I'm seriously impressed by the range of things there. So let me just see if we've got anything in the chat. I'm not sure we've got any questions at the moment. So does anybody have any questions that they would like to ask Henk Jan? Just see if anybody wants to open their microphone or anything.
I would have one.
Oh yeah, Fabio, go for it.
Yeah, thanks for the wonderful talk, Henk. Very amazing stuff you showed. I was wondering if among those studies of when colliding all these studies together, you and your team were looking for relationships between the diversity and the vertical gradient of the diversity and the export of material and organic matter from the surface to depth. Like, could you find like a positive or a negative relationship between deep sea diversity and the amount of organic matter that sinks down to the seafloor?
Oh, that's a good question. That's also, so we did not specifically correlate diversity with vertical flux. I mean, it's one of the open question, right? How does diversity relate to carbon flux? So I cannot give you an answer to that because we did not really specifically target that. But I think it is a necessary question, and that could also be answered as we collect more and more observations from these regions, collect them in databases, combine databases, maybe within JETZON, from the different size spectra as well, size fractions as well, and to basically approach that question. I think it's a very good one.
Thank you. Yeah, not an easy one to solve either, I suspect.
No.
So we also have a question in the chat from Matthew Woodstock. Matthew, would you like to ask the question yourself and just undo the mic if you'd like to?
Sure. Yeah. So you mentioned that during the eDNA for pyrosomes that you were able to detect them 3,000 meters deep. I was just wondering if they're actually that deep, or is that signature from their sinking feces, perhaps?
Yeah, that's a very valid question. with eDNA you cannot say what the origin is of the sequence, right? What we can say is that we also observed, we performed pelagios tows down to 2,500 meters, and we did see a dead and sinking pyrosome with like a handful of them. So very low numbers compared to the surface ones, but we did observe them. But you're right, I cannot say if the eDNA was from a carcass or from feces. So you can say that pyrosome material is exported down to 3,000 meters. If it's a fecal pellet or a carcass, is not known from the eDNA alone.
Great, thank you.
Okay, thank you. So one quick question from myself. I mean, I know virtually nothing about eDNA. I mean, I know there are issues in terms of timescales. So the question was, you know, is it more difficult to interpret eDNA for signals which are undergoing some sort of ontogenetic migration, or are the timescales long enough that it's not an issue?
So we have not done a study on vertical migration specifically yet. One way that it could, so the longevity of eDNA ranges from several hours to several days, basically, and there's still too few studies in particular on mesopelagic and deeper fauna to actually support this. One could expect that with daily vertical migration, if you have a fixed pattern, that eDNA is almost everywhere. So you would, if it has a higher longevity, if it's only a few hours, you may be able to actually pick up the daily vertical migration signal. So that would be one approach. eDNA longevity is very much subject to temperature. So the colder the temperature is, probably the longer the molecules or the eDNA stays detectable in the water, because of reduced bacterial activity. So one could also expect that in surface water, if it's warmer, there may be less eDNA from the deeper organisms that have migrated up. But this is very much, I mean, this is definitely subject to more research, and we know too little about.
Thank you. I mean, it seems an incredibly powerful tool. I guess it's just the understanding quite what the tool is telling you is, once again, not a trivial question.
No, and it's, you know, I think it's a very powerful tool. In particular, when you can combine it with an ecological question. But what is really needed are these reference sequences, because, for example, GenBank is only sequences on there is only what people upload basically there, and what is important, that you have your reference sequences, that you know that the sequence behind is really the identity of the animal. So that's important.
Right, well, thank you very much, Henk Jan. I say, if anybody has any other questions for him, then, you know, I'm sure he would be happy to answer any questions. So please do get in touch, and we will be putting his talk up on the YouTube channel later. So thank you once again.
Yeah, thank you for having me. Good luck, Fabio. Looking forward to your talk, and nice to meet you, Adrian. Bye-bye.
Thank you. An excellent